but now it's good morning, ladies and gentlemen. First, I'd like to thank organizing committee for for my delivering the keynote speech here today. It's my honor to share the data with you on our research funding in the past 12 years, titled Ongoing Journey Targeting Citrullinated Histone H3 and the Early Diagnosis and the Treatment of the Sepsis. So, sepsis is a critical organ dysfunction caused by uncontrolled response to infection. According to WHO's data from 2017, sepsis affected over 49 million people, caused 11 deaths. And you know how dangerous, how harmful the sepsis is. But so far, no specific biomarkers for early diagnosis of the sepsis, and no effective treatment for advanced sepsis. So sepsis seems like an illness, and to present like the imbalance, the inflammatory syndrome, but it's not just illness only, and to manifest this syndrome. I'm working in the department of surgery for a long time, so I know, and maybe most of you know, trauma injury and sepsis experience a same syndrome. I mean, systemic inflammatory response syndrome. I mean, SIRS in the early stage. Presently, no efficient method to distinguish one from the other. Later, the sepsis treatment delay or unnecessary antibiotics use. So when you encounter the patient trauma injury or sepsis in the clinic, as most of you may be a doctor, and we ask, what's that? How I should treat the patient? However, no efficient the method. Since they are have the same syndrome, but treatment differs greatly. Our journey, also my journey, start from there. Targeting citrullinated histone H3. I mean, CH3. Later, I will mention. So this CH3 is modified by a group of enzyme which we call the peptidyl-arginine deadenase. Later, I call PAD. PAD is a group of the enzyme since 5 isoform exist. I mean, PAD, namely PAD 1, PAD 2, PAD 3, PAD 4, and PAD 6. Only PAD 2 and PAD 4 are able to translocate it into nucleus. Histone H3 is a nuclear member, the protein. And there, this PAD 2 and PAD 4 catalyze the conversion of the histone H3 to cit H3. And this diagram illustrated how it works. You know, microbial infection, including the virus, bacteria, or whatever, and that infection will activate the pad. Activate the pad, I mean pad 2 here or pad 4, translocated into nucleus. There, citrullinated histone H3. The citrullinated histone H3, cit-H3, induce the chromatin decondensation. Then the nuclear membrane fused to the cell membrane. The cells break, release the nuclear contents into extracellular space with the, of course, cit-H3, which is called NATS formation or mitosis. Normally, small amount of the NATS defends the host against microbial invasion. However, accumulating evidence has shown excessive NATS can detrimentally affect the immune cell and the organ functions. cit-H3 is a trigger for the mitosis, and it is strongly associated with the immune cell death and the organ injury. We have found in the previous 12 years, this citrullinated histone H3, on one hand, at the cellular and molecular level, can bind to other neutrophils, cause more mitosis, created a vicious circle there, which we call the vicious circle because this circle is self-perpetuating cycles. There, cit-H3 and the NATS formation continually enhance each other. This circle. And on the other hand, the release the cit-H3 can induce apoptosis of macrophage, endothelial cell, and epithelial cells. So, block this vicious circle can reduce the cit-H3 level and also be an effective treatment strategy for sepsis. This is our hypothesis. In the past 12 years, our group has centered on cit-H3. And we achieved 3 major milestones. Let me show you what are 3. And first, we established the cit-H3 as the diagnosis and the prognosis biomarkers for sepsis. Second, we identified the cit-H3 not as only as a biomarker, but also as a mediator, participate in the pathogenesis of sepsis, and sepsis induced acute lung injury. Later, I will say ALI. And also, we discovered typical role of the pad. I just mentioned only 2 pad can translocate into nucleus. Pad 2 is one of the 2 pads. And there, they play an important role in the pathogenesis of the sepsis, and the sepsis induced the ALI. Now, let's go through the 3 main milestones in detail in our clinical journey. First milestone is citrullinated as a biomarker. We started the identification of citrullinated as a biomarkers in the animal models. We took 2 different models. One is the hemorrhagic shock model, that's a trauma injury model. And the other one is the endotoxic shock model. Some people call it the sepsis model, but strictly speaking, that's an endotoxic shock model. You can see from here, in the hemorrhagic shock, there are no detectable CTH3 was found. However, in the endotoxic shock model, we see the CTH3 come out. I mean, circulating CTH3 come out early, even like 30 minutes after give the IP injection of the LPS. And peaking at the 12 hours, lasted for 24 hours when the mice were dying. That means cover whole parasites. And we treat them, the sepsis, I mean the mice with the PAD inhibitor, which is here, that's YW356. And this treatment can significantly reduce the CTH3 levels, which means CTH3 is a specific biomarkers for the endotoxic shock through PAD activity. And we compare with the existing biomarker. Maybe most of the audience know, in the clinical, procalcitonin or PCT. How was that? When you do the comparison, this panel, that's I just repeated, I put it here. How about this? And we check the PCT in every time point. You can see not significant difference until the 24 hours, the PCT levels going up, and the treatment with the PAD inhibitor, that's a non-specific inhibitor. Nothing to do nothing should do for the PCT, right? But the treatment with the PAD inhibitor can significantly decrease the PCT, which means show against. Actually, we already know PCT, nothing to do, right? Even that treatment can significantly reduce the PCT level. So, put together, CTH3 is a specific biomarker. At least we think, at least our experiment show you. How about the patient? How about the patient with the septic shock? We collected over 160 patients with the sepsis. And you can find, here I labeled 1 hour. That means we just found that this patient suffered with the sepsis. And 24 hours, that's the day 2. And 48 hours, that's the day 3. And we took the non-infectious shock patient as a control, like a trauma injury and hemorrhagic shock patient as a control. Because we also took the healthy volunteer as a control. Among the 5 groups, 3 of them significantly, CTH3 levels went up. How about the PCT? Same samples from same group, you can see no significant difference among all groups. Again, I show you CTH3 as the specific biomarkers. It holds the three very important features. One is specificity, second is sensitivity, three is last long, which provide our clinical doctor enough time for the diagnosis, for the treatment. How wonderful it is, CTH3. I don't know. Let the data speaking. We also take the V2 sample of the patient and did another analysis. You can see here, we have the sepsis survival patient, sepsis dead patient. 97 of the dead patient, I mean 97, the patient, if the CTH3 levels went up beyond, above the 0.2 nanogram per ml, this patient will be die within 3 months. I like it. In summary, the CTH3 as a specific biomarkers because of it appears early, prolonged duration, specificity to the infection induced shock, and one more is the mortality prognosticator. As I show you the last slide, 97 patients, if their CTH3 go up to 0.2 nanogram per ml, the patient will be die in 3 months. Okay, maybe you will ask the question. I already seen something. People use the CTH3 antibody. They found this CTH3 even appeared in the advanced cancer. I will tell you why. Please pay attention and also be patient, please. Second milestone is CTH3 as a mediator. I will tell you some interesting story. There, I will show you why some group got the CTH3 and different from our results. And I hope this story can draw your attention and give you a free to make your own conclusion. Our hypothesis at that time was neutralize the circulating CTH3 with a specific monoclonal antibody against the CTH3 so the improve survival. With this hypothesis, we bought a CTH3 antibody from market. How was? Unfortunately, we found no specific surviving improvement. We asked the question, why? What went wrong? Theoretically, it should, but it shouldn't. It wasn't. We are not the only group asked a question. We are not the only group disappointed. And Dr. Li and Dr. Reddick, they published a paper in 2016. The title is here, Current Challenges and Limitations in Antibody-Based Detection of the Citrullinate Histone. They found some commercial antibodies to detect the CTH3 called jeopardize. Wow, this word is very serious. Jeopardize effort to reach scientific consensus. They alert us, please take cautions to the interpretation of any single antibody for determination of pad activity, including pad 2 and pad 4. And they strongly suggest needs a new CTLA-3 monocantibody. So we plan to make a new CTLA-3 antibody. But first question, we have to think about what's the antigen we should use. And we have a published paper and show in the N-terminal sequence of the histone H3, there are several cleavage sites. One of the cleavage sites at 728 by Jackson L. So we think the well-designed antigen should cover this site. In addition, in the N-terminal sequence of the histone H3, there are 4 arginine can be converted to citrulline by 2 different kinds of enzymes. One is the PAD4 and convert the arginine 2A17 to citrulline. Another enzyme is the PAD2 and convert the arginine 26 to citrulline. 2 different antibodies, both of them can translocate into nucleus for their activity. So well-designed antigen should cover both PAD2 and PAD4 catalyzed citrulline. With these antibodies, we develop a new CTLA-3 antibody. That's I would say novel CTLA-3 antibody because nobody used that so far. And we compare the 2 antibodies. One is our own antibody I call the CTLA-3 monocantibody 4C. The other one is the commercial antibody CTLA-3 monocantibody 3C. You can see the Western blot analysis show you, and both of the antibodies specific, nothing to do with acetylated H3, nothing to do with the methylated H3, nothing to do with the unmodified histone H3. But our antibody appeared to show stronger signals. Maybe you will argue, this is just a Western blot. Okay, I'll show you the ELISA data here. With the new antibody, antibody 4C, I can detect a greater amount of the circulating CTLA-3 compared to the commercial antibody 3C, just pick up 1/3 of the circulating CTLA-3. We confirm that with the gene knockout. See here, knockout as either PAD2 or PAD4 cannot abolish, complete block, complete suppress the circulating, the CTLA-3 production. However, knockout both are called the double knockout. Double knockout the PAD2 and the PAD4 abolish, completely suppress the CTLA-3 expression. Okay, then with the new antibody, we did an important in vivo survival improvement experiment. You can see here the red dot compared with the blue lines. The red dot is the new antibody. The blue line is the commercial antibody. The new antibody can significantly improve survival compared to the commercial antibody to nothing. And we did a further study. So we take the antibody and inject the in vitro IV injection of the antibody, and we found the new antibody can abolish the net formation. You can see here, compared to IgG only, that's a baseline. But the commercial antibody cannot. So what's the mechanisms involved? At that time, we asked a question ourselves, and we did an in vitro study here. We treated the freshly isolated neutrophil with CTG3 peptide. That's a 30 N-terminal 30 amino acid. And also take the Ag3 peptide, that's an unmodified 30 amino acid N-terminal peptide. And we incubate with the neutrophil. You can see CTG3 peptide can stimulate the net formation. Here, merge, overlay, but the Ag3 peptide did nothing. This suggests that the peptide, the N-terminal of the CTG3 30 amino acid can somehow induce, or you can say stimulate the net formation. And here you may, this data suggests that there is a vicious circle there. I mean, perpetuating, self-perpetuating the net formation. CTG3 net formation enhance each other continually. Okay, it's not enough. We did the in vivo study, and we inject the same peptide IV injection into the mice. And here the data show you surprising result. First, CTG3 peptide just 3.5 kilodalton and can induce the 17 kilodalton CTG3 protein production, which confirm our concept, vicious circle there. And the second interesting result is CTG3 peptide. This induce the 17 kilodalton CTG3 protein expression is accumulating in the lung, but not other organs like heart, liver, and spleen, kidney, brain, and intestine. It strongly suggests the pivotal role of CTG3 involved in the sepsis-induced acute lung injury. Okay, you may argue this is just your hypothesis. Here, I show you something more. We found the CTG3 levels are increased in the plasma and BAF, bronchial alveolar lavage fluid of the patient with the sepsis and the ARDS. And we assess if this CTG3 increase is due to the sepsis-induced ARDS or ALI. And we did a mouse studies. And here we take the CRP-induced CTG3. Maybe most of you know CRP, that's a Seeker's ligation and puncture. That's a typical CTG3, typical sepsis mouse model. And this model can induce the CTG3 increase in the blood serum and in the mouse lungs tissues, which suggests CTG3 may contribute to the sepsis-induced ALI, or you may say sepsis-induced ARDS. And at that time, we asked, okay, since the CTG3 can do that work, how about you neutralize the CTG3 with your new CTG3 antibody? Here is. We found that the CTG3 new antibody can significantly improve survival in the lethal mouse sepsis models, as shown in the panel A, and decrease the sepsis-induced, ALI, as shown in panel C. Mechanism studies shown in panel B. The new antibody can reduce the cleave the caspase-1. You can see here that's a T12. See the two band here. One is the control, the other one is the antibody treatment, and also reduce the cleavage IL-1 beta. This is the feature of the process. So this result suggests or indicate this new antibody works well and did the work we expect, we hypothesized. In summary, CTG3 forced monoclonal antibodies recognize PET2 and PET4 citrullinated CTG3, while your commercial antibody 3-CT only recognizes PET4 catalyzed CTG3. CTG3 4-CT antibody peptide stimulated CTG3 protein production both in vitro and in vivo, which the block, perpetuating a vicious circle, and then neutralize the circulating CTG3 with the, from the CRP mouse model, and reduce the mortality and diminish the caspase-1 induced ALI. Now let's move to the third milestone. We discovered PET2 play a role in the sepsis pathogenesis. PET2 and PET4 both are PET enzyme translocated into nucleus, but for many years, research primarily focused on the PET4. Inhibition PET4 can protect the immune cell death and prevent the sepsis-induced ALI. Our data strongly suggest PET2, not PET4, play a role. For any people, in case you want to argue with me, let's do the comparison. We took the PET2 knockout mice and the PET4 knockout mice, and we did a comparison. PET2 knockout mice can improve survival. You can see the left panel, but not the PET4 knockout mice. And we also did a comparison. We found the PET2 knockout mice can ameliorate acute lung injury in the Pseudomonas aeruginosa induced sepsis and sepsis ALI. You can see here, I would like to draw your attention from this part. You can see clearly see the macrophage there, right? But not the wild type mice. And this panel, that's ARI scores. We send the HE staining slides to the board certified pathologist, and this is blend, and they make a score for us. You can see here, type 2 knockout mice can significantly reduce the ALI score. That means protect the lung function. But not the PET 4. Now macrophage you found here, and the ALI score no significant difference compared to the wild type. And we also took the 5 cells and did the flow cytometry assays. So you can see the PET2 knockout. You can see a lot of the macrophage there, but not the PET4. And PET2 knockout can decrease the pyroptosis in the bone marrow-derived macrophages. And you can see here, and decrease the cleavage caspase 1. See the 2 band here. One is the wild type. One is the PET2 knockout. Can see the column here significantly, but not PET4. And also, this is not enough because the main feature of the pyroptosis not only the caspase 1 cleavage, but also should see the IL-1 beta secretion and the IL-18 secretion. You can see here, and the PET2 knockout significantly and decreased IL-1 beta and IL-18 are also the LDH, I mean the lactate dehydrogenase, and not PET4. And see here, nothing changes compared to the wild type. And functional studies show the PET2 knockout can enhance the bacterial clearance from lung, from bath, from blood, from spleen, but not the PET4 knockout. Here, in summary, PET2, but not PET4 knockout offers a protection against the immune cell death and ALI, I mean acute lung injury. PET2 knockout, but not PET4 knockout, increase macrophage count, decrease pyroptosis, and enhance function of the bacterial clearance. I'd like to make a conclusion here. First, citrullinated serve as the diagnostic and prognostic biomarkers for sepsis in both mice and humans. citrullinated offers a therapeutic target for sepsis and acute lung injury. And commercial citrullinated monoclonal antibody has a research limitations there. I give you the answer why some groups say citrullinated also a biomarker for cancer, because they forget about the PET2 catalyzed arginine 26. The new citrullinated monoclonal antibody overcomes the shortcoming the commercial antibody said. And the PET2 represent a promising treatment target for sepsis and acute lung injury. And blocking PET2, not PET4, please. CTG3 pathway is of our future research direction. We are currently continually our journey to advance. And I like to take these figures we recently published in Frontier Immunology and tell you what's the future direction. And see, when the bacterial or microbial infection activated the PET2, activated the PET2 translocated into nucleus, and citrullinated histone H3 there, and make the mitosis, release the CTG3, and this release the CTG3 can bind to the membrane receptor. Sorry, please forgive me. I cannot say much about that because our lab is currently doing that. And the bind there specific receptor and in cause more mitosis, and that's the service circle. And mitosis and the service circle. And this release the CTG3 also can bind to the macrophage by paracrine and induce the parotosis. So our aim is we seek block the PET2, I mean inhibit the PET2. We have the specific inhibitors with my collaborator from the UMass Worcester Medical School there, and block the vicious circle. This is our direction to protect the immune cells from death and prevent the sepsis and ILDs. I'd like to thank the funding agent for their financial support. I'd like to thank all the people I listed here. Their dedicated work make my presentation here possible. And also, I'd like to thank the University of Michigan Patent Office and the University of Virginia Patent Office for the patent, approve the patent and the current still in the provisional patent. And thank you all the audience for your attention. I'd like to stop here for any question you may ask. Thank you.